Growing plants from seed is one of the easiest and best ways of producing large numbers of plants. This video looks at seed collection, conservation, storage and treatment. Seeds may be purchased from nurseries and companies which produce true-to-type lines of selected species and cultivars. A true-to-type line is one which is either a straight species or one that has been hybridized in such a way to guarantee that all seedling plants will produce the characteristics that are desired. These characteristics may include flower colour or size, plant habit, disease tolerance or other characteristics. This seed is sold in sealed packages, usually with guaranteed germination rate. These specimens are guaranteed that under appropriate germination conditions they will produce at least 90% germination. Seed catalogues provide further information on the germination requirements of particular species.
Seed may be coated or palletised. Specialised machines apply the chemicals to the seed accurately and evenly. This may be done for a variety of reasons. Seedling nurseries like palletised seed because it makes the seed a uniform size and is therefore much easier to use in an automated production system like this vacuum seeder. Other seeds are coated in plant protection chemicals such as fungicides or insecticides to minimise storage and germination problems. In the case of this seed, the pink coating contains the fungicide. Some seed is also coated in a small amount of fertiliser to aid growth after germination.
In recent years, Landcare and bush regeneration groups have planted millions of trees. One of the groups involved is Greening Australia. Simon Conlon from Greening Australia explains some important concepts in gathering seed for bush regeneration.
The first thing that we consider is collecting local seed for the local project. This is known as provenance. It's important to source provenance seed because it helps to maintain the biological diversity for our projects and genetic diversity as well. This is aimed at conserving the native flora attributes of the site, and also plant performance is increased due to these local varieties being used in the local project for local conditions. Moreover to that, it maintains the local character of the area, and also this provenance overall allows for the success of the project. If the wrong provenance is planted, it can lead to disastrous effects on your project. One example of this is Moreton Bay ash or Eucalyptus tessellaris. It grows in coastal areas from Brisbane to Cairns, but it also grows in western areas out around Charleville. If you were to source seed from the coastal area and plant it out in that western area, you'd probably have disastrous results due to the fact that that seed hasn't got frost tolerance and drought tolerance built into it. When you're out in the bush collecting seed, normally you come across three different types of seed. You've got grass seed, fleshy fruits and dry fruits like eucalypts. One important thing when collecting grass seed is to collect mature seed. We normally do this in the field by stripping the seed off the head easily. That means it's dry and ready for storage. Another large-scale way that we collect grass seed is with a mechanical harvester. This we use for large-scale projects that we need large volumes of seed for. One common dry fruit that most people would know is eucalypts. We normally collect that by using special devices such as a thrower to get the fruit down from high up areas, and from there we also sometimes have to ascertain whether the seed is mature before it's opened. We normally do this by the cut test. Other dry fruits such as Kurrajong are mature when they open and they hold their seed for a long time. Fleshy fruits such as fig species, we normally gather those by checking to see if they're mature by their colour. They normally change colour from a green to another colour like yellow or red. And then also checking for the feel, the consistency of the fruit. If it's soft, it normally means it's mature as well. Whichever seed you collect, it's important to make sure that you get the right provenance and the right maturity. The other important component is to make sure that you've covered legal requirements by doing a bit of research in the Nature Conservation Act and Environmental Protection Act.
After collecting or purchasing seed, it may be important to see if the seed has a chance of germinating. This is called viability testing. There are four commonly used viability tests: a cut seed test, germination sampling, a float test, or tetrazolium testing. The cut seed test involves taking a seed making a cut towards the centre and checking to see if internally it is firm and fully sized. If it is, the seed is quite probably viable. This is an example of a seed that looks fine. This seed, on the other hand, is an example of a seed that most certainly has died and will not germinate.
The germination sampling test is commonly carried out to test the germination rate and viability of seeds before sowing. For very fine seeds, it's more practical than the cut seed test. A sample of the seeds to be tested is measured by either weight or seed number. The seeds to be tested are germinated either in an incubator or on a propagation bench. The actual number that grows can then be tested against a standard such as the typical number of seedlings per gram or milligram. The float test simply involves placing seeds into water. Those that sink are probably viable and suitable for sowing. Those that float are probably dead and need to be discarded. The float test is not foolproof because some plants naturally use water to help disperse its seeds. Therefore, some viable seeds will float. The final common test is more typically carried out in a laboratory, the tetrazolium test. Dr. Mark Sutherland is a plant physiologist from the University of Southern Queensland.
You'd use the tetrazolium test when you needed to find out quite quickly whether or not a sample of seed was viable. The test will give you an answer within 1 or 2 hours. Tetrazolium testing begins with making a dilute solution of the tetrazolium compound in water. Then all you do is take a sample of seeds and soak it for a couple of hours in this dilute solution. If you've got really large seeds like maize, you cut them in half to aid penetration of the solution into the seed. What happens is that living tissue reacts with the tetrazolium to change it to a red-coloured compound. So that what happens in a living seed is that the embryo, over a period of time, stains dark red. You can then count in a sample of seeds how many have red-stained embryos, and that will tell you what percentage of the seeds are still viable in the sample. Viability testing is really important because, first of all, commercial suppliers of seed need to be able to guarantee that the seed they're selling is viable and of high quality. Secondly, people that use seed, such as nurseries, bush regeneration groups and land care groups, need to know that the seed they're buying or the seed that they've stored is still viable.
If seed is to be stored for some time, it is important that the seed is stored correctly. The first step is to make sure that it is correctly labelled. The information should include name of the species, date of collection, location of the collection, and viability test results. Most seeds can be satisfactorily stored in a cupboard or a box in a dark, dry area with a stable temperature. A box like this is fine for many species. However, for optimum storage conditions, a fridge or a cold room can be used. This commercial nursery uses a cold room maintained at between 6 and 8 degrees and at a low humidity of around 20%. It is very important that the seeds stored in a fridge or cold store are in sealed containers or packets. Seeds should be stored in a location safe from children, as many seeds are poisonous. When it is time to sow seeds, there are a number of requirements that must be met for the seeds to have a chance to germinate. Seeds need appropriate levels for the species of water, oxygen and temperature. These are provided by an appropriate growing media and suitable conditions. For most species, light is not required for germination, but light is always required shortly after the seedlings have emerged to allow the process of photosynthesis to begin. For many seeds, if the right conditions are provided, the seeds will germinate with little fuss. For other species, some form of seed treatment may be required. To know what sort of treatment is required is not easy, so you may rely on research, from discussion with other propagators, or from your own experience.
Scarification is a seed treatment using some physical means like a hammer or sandpaper to damage the seed coat. After the hard seed coat is broken down or damaged in some way, water can be absorbed and the young embryo can find its way out. This process of scarification is commonly used with hard seeds like acacias. Some seeds germinate far better after a period of wet heat treatment. For example, treatment with boiling water. Some seeds require a period of dry heat, such as placing in an oven. In both cases, the heat helps to break the seed coat down, allowing for the seed to germinate.
Germination is promoted in many plants from countries such as America, South Africa and Australia by exposing the plants to smoke. There are many thousands of chemicals in smoke. Some of these chemicals seem to have a role in mobilising the energy that is stored in the endosperm, that is, the food store in seeds. This energy is then used by the developing embryo to grow towards the light. To provide the suitable conditions for germination, these seeds may be placed in a smoke chamber and left for a period of time. Alternatively, a product called smoke water is available for use in nurseries. This product is diluted in water and the seeds soaked in the solution prior to sowing.
Some seeds have a fleshy outer covering. In many cases, this flesh contains chemicals that stop germination. In nature, these plants would be exposed to a prolonged period of wet weather prior to germination. Therefore, the flesh needs to be fully removed before germination can occur. This may involve soaking for a period of 24 hours or longer, or placing the seeds under running water. This process leaches the growth inhibitors. To save water, some propagators place the seeds in a porous bag and place that inside a toilet cistern. This saves water and prepares the seed. Some seeds need a period of cold chilling prior to germination. The term to describe this treatment is called stratification. This applies more to plants from cool climates, such as some southern species and many exotic garden plants from temperate regions. Seeds are stored in a fridge or cool room for a period of up to 3 months prior to germination.
Collecting and growing seeds for regeneration, pleasure or profit can be most rewarding. The techniques shown in this video are an introduction to what can be anything from a simple to a most sophisticated process of growing plants from seed.